Yeah, sure. We're FateTx is a cell-based cancer immunotherapy company. And for the past, almost 7 years now, we've been pioneering a technology called induced pluripotent stem cells, or iPSCs for short. And the overall vision of the company is really quite simple but profound. The goal is to make cell therapy look like monoclonal antibody therapy. So much broader patient access and do it much more cost effectively. And we use iPSC master cell lines to allow for mass production of NK and T cells. The utilization of iPSCs allows us to do complex engineering, and that we can embed within cells multiple levels of anti-cancer functionality. And again, we do this at the stem cell stage. And we create a master cell bank that allows or enables the ability to create thousands of doses of NK or T cells. So we can do that very cost effectively, and that does help us deliver to patients much more broadly cell-based cancer immunotherapies. We have a very large pipeline of NK cells that are already in clinical stage of development. And we really like T cells as well. It's just taken us a bit longer to figure that out. But we do have a program that's T cell based in the clinic, and we're working on multiple T cells with our collaboration partners, Ono and Janssen Pharmaceuticals. And then lastly, I would just say that clinical pipeline is directed at both liquid and solid tumors of interest. And so we're super excited. It's been a heck of a journey over the last 5 to 7 years, but that's in a nutshell what the company's hoping to do.
Yeah, that's great. And I mean, I think you guys clearly sort of pioneered the iPSC space, but you're also sort of not the only game in town anymore. So I wanted to ask, with more companies working in or at least showing interest in iPSC-derived cell therapies, what does Fate's competitive moat look like in that area?
Yeah, it's there's a lot more competition. I'd say a few things. Look, we have a really strong intellectual property positioning. We've got, I think, more than 350 issued patents and about 150 that are on file. And so we have a really strong IP moat. We'll utilize that at the appropriate point in time. So I think we feel really good about our intellectual property position. There's just a lot of know-how, Matt. This stuff's really hard to do. I could give you the recipe of how to do iPSCs. It would take you a long time to figure it out. So we've learned a lot in the journey over the last 6 or 7 years since we pivoted the company in this direction. And then the third thing I would say is there's a lot we as a company do not know, but we as a field of cell therapy and NK cells in particular that we do not know. There is a lot of value of taking concepts, getting through preclinical development, and then ultimately getting into patients. And we now have 5 or 6 different programs in different tumor types. And we have a lot of translational insights that the company is gleaning over time. That helps us in many ways. It helps us more thoughtfully develop our programs in the clinic. It also helps us identify areas for new innovation as we think about the next generation of programs. So I feel like we're in a really strong position. We are pretty far ahead in the clinic with multiple different programs, and I think that benefits the company. But we are clearly pioneering something, and it takes time, and we're learning lessons along the way. But I feel like we're in a really strong position from IP all the way through experience and know-how.
Yeah, and you kind of touched on it, and I want to dive into the pipeline. But first, I want to ask sort of a big picture question, which is just, where do you see Fate's technology in 5 to 10 years? And where do you ultimately hope to see Fate's therapies within the standard of care of various cancers?
Yeah, we could talk a half an hour on this alone, I think. I'll try to keep it simple. There's 2 or 3 things that come to mind. iPSCs, they're great for many different things, but they also can be differentiated into more than 200 different cell types. So while today we're focused on NK and T cells, you could generate just about any cell type in the human body. So you can start thinking about things outside of oncology. You could think about regenerative medicine. You could think about autoimmune disease. There are a lot of companies applying now iPSCs outside of oncology. I think we'll end up exploring some of those options as well. So I think there's super exciting potential there. I think closer to home, though, we do make NK and T cells. We know how to do that really well. Where can NK and T cells go outside of oncology? I think there's some near-term opportunities that we can explore just with NK and T cells. And then sort of the 3rd or 4th things I like to think about. We are now looking at NK cells in isolation, and we're now beginning to develop T cells in isolation. And I know most companies are either NK or T cell focused, but Fate's somewhat uniquely positioned in that we do both. And if you think about your immune system, it's the combination of many different cell types. So why not combine NK and T cells, innate and adaptive immunity, and might that cause some interesting clinical benefit to patients? So I think we're positioned to think about doing that in the near term as well. And then maybe even more tangibly, the 4th thing I would say is our products, and if you look at our NK cells in particular, they are designed to synergize with monoclonal antibody therapy. So whether that's in lymphoma or multiple myeloma or in solid tumors, there are notable regimens, including monoclonal antibodies, that represent standards of care. Our ability to simply add on top of those therapies is really compelling at multiple different levels. And we're super excited about being able to do that initially in relapse refractory patients, but working our way up into the treatment paradigm, including very early line settings. So I think we're super enthusiastic about iPSCs in general for all those different reasons.
Great. Yeah, I want to talk about all of that, but maybe we can dive into the pipeline now. Starting with lymphoma, we saw a lot of new data at ASH from FT516 and FT596. First, I just want to ask what were sort of your main takeaways or learnings from those trials, and how's that informing your strategy going forward in lymphoma?
Yeah, big picture, I think a few things. One, we're starting to see some interesting responses in indolent lymphoma, in aggressive forms of lymphoma, And in both patients that are naive to CAR T-cell therapy, and what is very differentiating is downline of CAR T-cell therapy, whether you're talking about FT516 or FT596, roughly about a third of our patients have had the benefit of prior autologous CD19 CAR T-cell therapy. We're seeing responses, including some durable responses in those patient populations, and it looks like a potential fast-to-market opportunity. So we're seeing responses across the board, indolent, aggressive, pre-CAR T, post-CAR T, and I think early signs of durability, which I know has been a clear focus for in the investment community as us and clinicians as well. So we feel like we've got fast-to-market opportunity. We are now progressing into expansion studies with FT516 in different cohorts, and we can talk a little bit about that. But the company also benefits now from RMAT designation for FT516 that we received earlier this year. So we have the ability now to go in with the FDA, talk about our plans and strategy, and begin to work towards what we hope is an initiation of a pivotal study by the end of this year.
Yeah, that's exciting. And you mentioned durability. I want to ask a little bit more about that. It seems like there's a temptation to sort of compare NK cell therapies directly to approved autologous CAR T therapies. But I mean, is that really fair? Or I guess, how should investors be thinking about durability of response with, Fate's programs and other off-the-shelf, cell therapies?
Yeah, there's not a simple answer there. I get that we're cell therapy and people want to make the comparison. I do think setting matters quite a bit, right? So if the conversation, are you competing head to head against, Yescarta in an academic center or institution, I think it's a fair comparison, right? We are running those studies, including now an expansion with 516. And so if that's the ask, can you compete with autologous CAR T-cell therapy? I think that's the right benchmark to think about. For our initial fast to market and for other opportunities, either in the community setting or in early lines of therapy, clearly that's not the right benchmark. In fact, there is no standard of care now downline of CAR T. So while that's been a very good outcome for patients that can receive that therapy, the question of what do you treat those patients with next is an unknown. And we've seen some data at ASH from a couple different consortiums that show, complete response rates for checkpoint inhibitors and other physician choice therapies, is in the range of maybe 10 to 20%. The PFS is very short, 1.5, maybe 3.5 months. And overall survival, that's less than a year. That's not a great proposition for a patient downline of CAR T. And we think that's a meaningful opportunity for us with either 516 or FT596.
Yeah. So with that, you mentioned, 516 just got RMAT, but 596 is sort of the more highly engineered therapy. So I'm sure you're asked this a lot, but how are you thinking about FT516 versus FT596 as those programs progress? And is there a chance for 596 to sort of leapfrog over 516? Or, how are you thinking about that?
Yeah, this portfolio strategy, rationalization, prioritization question comes up quite a bit. Look, 516 is simply further ahead, right? We've built our platform modularly. And just to remind people, FT516 has, a CD16 receptor that's designed to combine, in this case, with rituximab. So we really like the biology there. It's played out really well. We're seeing the responses that we sort of alluded to. FT596 does now include a CAR to CD19. So this ability for a cell to deliver multiple levels of functionality is important. I think we're big believers in dual or multi-antigen targeting. So I think preclinically you see 596 having advantages. I think clinically there may be some early signs that that's the case. But that said, we are still completing dose escalation. And as you know, we've only more recently begun to dose in what I would consider a more optimized fashion with 2 doses per cycle. So I think we have more work to do with FT596, but we are working very hard to close that gap. And as we think about planning, although RMAT designation is clearly for FT516, that playbook, if you will, in aggressive lymphoma downline of CAR T could apply equally to either program. And so we may be in a position and hope to be in a position and are looking to be in a position before the end of this year to have enough data with 596 to potentially declare that as what we think is going to be our best-in-class program and the ability to run with that program and that program alone in B-cell lymphoma. But I think we need a little bit more data to make those determinations.
Yeah, sure. And so I guess in the meantime, I want to ask about the expansion cohort strategy with FT516. And I guess to start, could you speak to the evidence out there in support of preconditioning with earlier line chemo as you're starting to look at with 516 and also I guess down the road too with 596 with a R-CHOP?
Yeah, so one of the expansion, there's 4 cohorts that we can talk about with FT516. The one that's relevant for this question is the combination with bendamustine and rituximab. Mm-hmm. And I think there's 2 sources of support I would suggest you look towards. One is simply Novartis' Kymriah. If you look in their label today, about 20-25% of the patients, is my recollection, use bendamustine as the conditioning regimen as an alternative to Cy/Flu. And as part of their ASH update just last December, their next generation program is also providing for the use of bendamustine as a conditioning regimen. So I think that's point one. The second one is there's actually an interesting poster from ASH, again, last year in 2021, out of University of Pennsylvania. It was about a 135 patient study. And the investigators looked at, prior to Kymriah therapy, utilizing either bendamustine in 2 doses, I think 90 milligrams per meter squared, typical dose of bendamustine schedule, versus Cy/Flu in a typical 3-day lymphoconditioning regimen. And it was 2-to-1 randomization. So you had twice as many patients, about 90 in bendamustine and about 45 in Cy/Flu. And I'll cut to the chase, but what you saw is no differences in response rates to Kymriah. You saw no differences in PFS. In fact, numerically, the bendamustine arm was a little bit higher, but non-significantly so. But what you did start to see is bendamustine was a safer, more well-tolerated regimen. So you had lower rates of ICANS, lower rates of CRS. You had a lower amount of hospitalization stays. And so no impact on efficacy, probably a numerically slightly better profile from a safety perspective, but seems to be a very effective lymphoconditioning regimen. So I think we're interested in exploring that, in part because bendamustine is a standard used in earlier line therapies, and it's combined with rituximab, which our cells are designed to synergize with. So we're excited that that escalation arm is, or expansion arm is underway, and we'll hope to share data from that probably towards the end of this year. Just briefly, you mentioned another early line opportunity, this with FT596, and we're in the process of writing the protocol and hopefully submitting that to the FDA soon, and that's to combine with a frontline standard of care known as R-CHOP. And so there's less data there. R-CHOP's been a standard of care for 2 decades, but as it relates to its ability to effectively lymphoconditioning, that's sort of a lot in what is lymphoconditioning used for. There's less known there. But I think there is clearly immunosuppression when you give chemotherapy cocktails. You would see that, and you expect to see that with R-CHOP. So again, similar pattern. It's given in 6 cycles. Our ability to just add on top of that is existing standard of care. I think it's important that we tease that out, and we'll look to do so starting this year.
Great. So I guess, 4 expansion cohorts with 516, dose escalation with 596. When or what should we expect in terms of updates this year from the lymphoma franchise?
So we try to be as transparent as we can. And if you recall last year, in anticipation of cutting the data for ASH abstract submission, which happens relatively early, by the middle of the year. So last year we did an end of June, early July data cut across our programs for the purposes of submitting those ASH abstracts. We then had an opportunity to provide investors with an update, which I think was in August. So sometime in the third quarter, you would expect something very, very similar as we prepare for ASH. So we'll give an update likely in the third quarter on both programs. And then as we get to ASH, we'll have an opportunity to give a fresher update on those programs as well. And so you'll see dose escalation data for 596. You'll see the dose expansion data for FT516. And we may get to this map, but we also have the T-cell program that we've enrolled patients and cleared the first dose level called FT819. And investors should probably expect an update by the end of the year around ASH for that as well.
Great. Yeah, so great. So I actually want to shift now to AML. And you guys had some updates last year, I think, from 12 patients with pretty respectable 30% CR/CRI rate, but it seems like maybe the street doesn't give as much credit to that program as the other franchises. I guess I wanted to ask maybe any thoughts as to why, or what do you see as the main hurdles to overcome with that AML program?
Yeah, it's been almost a year since we've given an update, and my recollection of the data with 516 specifically at least 90 and 300 million cell dose levels, we saw 4 to 9 patients have a response. Look, AML is a really tough disease in multiple relapse refractory patients. It's a disease of the bone marrow. You not only have to clear tumor cells, you actually have to have recovery of the patient's hematopoietic system. That is not easy to do. That's a very tough ask. So the fact that we've seen any responses, I think, is very, very encouraging. 2 of those responses out of the 4 that I mentioned, my recollection is, are at least at 6 months, if not longer. So some signs of durability, and again, a really challenging disease. If I put my commercial hat on for a moment, it's not the same size as something like lymphoma or myeloma or certainly solid tumors. And so in some ways, maybe relatively less interesting for some. And I think there's been a lot of NK cell usage historically, sort of patient or donor derived, sort of non-engineered cells. And you've seen this sort of 20, 30% response rates. They seem to be very, very safe. And I think our cells sort of align with that. I think AML, unlike the other areas that we've been exploring, it doesn't have a standard of care monoclonal antibody that we can easily combine with. So in many ways, these engineered features, whether it's 516 or 516, aren't really being employed because we're not able to combine with a monoclonal antibody. So I think that's sort of the rub here until we find an antibody that is safe and effective that we can utilize to combine with. And I think we'll look for that in the future.
Yeah, I mean, on that topic, I know you guys actually just started an investigator-sponsored trial with daratumumab in AML. So, I mean, is that an opportunity to sort of improve targeting in AML? In your view? It
is, yeah. In the literature, there are lymphocytes that express CD38 that are upregulated. And so the thought of combining daratumumab with a product like FT516 or more likely for us now, FT5 38, which is what we're doing in an IIT with the University of Minnesota, is attractive. So that study is ongoing. We will likely give an update in the AML franchise sometime in the second half. I think we want to complete dose escalation for FT538, but we should then have a good data set to compare 538 to 516 and potentially share the first data from 538 in combination with daratumumab.
Gotcha. Well, maybe we can shift gears to multiple myeloma. And I wanted to ask, it seems that, naked or unmodified NK cells have maybe struggled more in myeloma than in AML or lymphoma. So I wanted to ask if you agree with that. And if so, why do you think that's the case? And then do you think 538 or 576 can overcome some of those limitations?
Yeah, I'm not aware of a huge amount of data with sort of unengineered NK cells. There's a lot in AML and MDS. So, myeloma, I don't think there's been a whole lot of exploration. In general, though, as you know, it's a disease that responds well to multiple different mechanisms of action, right? You have triplets, quadruplets that are in play in multiple myeloma. So as you think about our ability to intervene, FT538 is an optimized NK cell backbone, and it's designed to synergize with monoclonal antibodies. Of course, daratumumab should be the first thing that comes to everyone's mind that are familiar with that treatment landscape. There's a really strong, compelling reason to combine with daratumumab, and we're doing that phase 1, early days, first dose level, but we're in dose escalation in that combination. FT576 is a lot like 596 in that it allows for dual antigen capability. So in addition to synergizing with CD38 targeting monoclonal antibodies, we've introduced a novel CAR targeting BCMA. And so I think there's a lot of value in going after multiple antigens. And our hope, and you may have a line of questioning around this because we get it quite a bit, is likely lymphoma, maybe we can rescue patients, including those that have already experienced some sort of BCMA targeting therapy. So we have initiated our phase 1 study with FT576. And the hope is we could have a similar playbook start in late relapse refractory patients, including those that may have been prior exposed to CAR T containing BCMA regimens, and then move our way up into the treatment paradigm.
Yeah, I do want to ask about that because we have seen some recent approvals in autologous BCMA targeting CAR T. I also want to ask, just the same sort of situation as with lymphoma where 538 and 576 are both advancing through development. But it seems like they're a little bit closer together in terms of where they're at. So again, how are you setting these programs up relative to each other? And could 576 progress maybe a little bit quicker than 596 did in lymphoma based on the experience you've had so far?
I think so. Admittedly, 538 was a little bit held up. The way myeloma worked is that we had to clear the first dose level in AML, and we had about 7 or 8 screen fails in that first dose level of AML, which really slowed things down. So they are much closer in time and space, as your question rightly points out. Keep in mind, these things become a little bit more obvious in today's sort of mindset. 2 or 3 years ago, we'd have any idea if NK cells were a thing. And as we've modularly built the platform, things like FT538 represent a nice evolution in our thinking. And so, again, it's got the CD16 biology that we've talked about. It's got the IL-15 receptor fusion to fuel the cell. It also has the CD38 knockout, which was originally designed for multiple myeloma purposes, so we don't have fratricide associated with daratumumab utilization. What we've actually serendipitously found is that actually creates a much more metabolically fit cell. It just does better even in really scary sort of high oxidative stress environments. And so we want to test all those features in that biology. So I think it's important that we do FT538. Our goal with 576, though, is to make sure that our BCMA binder really works. It is novel. It's designed to bind to very low levels of BCMA expression. And if you've been following the BCMA landscape, unlike CD19, the binder seems to really matter. You can get very different outcomes in PRs and CRs depending on the binder. We want to make sure our binder is as good as we think it is. It looks great preclinically, but we want to test that out. And until we have better comfort at, probably medium to higher dose levels, it makes sense to run these in parallel. There's a lot of lessons to learn. But I think the possibility of consolidating to FT576, should it be safe, should it be effective, should our binder be very good, I think is a real possibility, but we probably will get there in 2023.
Sure. So I guess this year, what should people be expecting from the myeloma franchise in terms of updates? I don't think you've previously shown anything. So So looking forward to hopefully some first-in-human data.
Yeah, as are we. We have not shared anything to date. It's pretty early, 538, very early, first dose level. FT576 even earlier. So I'd like to see, the first couple dose levels if we can get there by ASH with FT538. So that's probably the likely point where we give an update. And then similarly, probably the first handful of patients of FT576, which again is already underway. I think we should be in a position to give an update around ASH as well. So I think that'll be the first time we share data from those programs.
Sure. Okay, so maybe we can move on to solid tumors. And I guess an update last fall from FT516. Are you using that more as sort of proof of concept, or what did you learn from that trial that read out back in I guess November it was?
Yeah, we had FT500, which is sort of an unengineered iPSC cell, and then FT516, which I think everyone is aware that solid tumors is a much bigger hurdle, a much higher ask. It's a much more challenging set of tumors. So those are initial foray, right? You're trying to answer the questions: can you administer NK cells safely? In the case of FT516, is there any indication that you can combine with monoclonal antibodies, like avelumab or ADCC competent, and see sort of any clinical benefit? Are they safe? A lot of the take-home lessons around translational insights, just understanding from tumor biopsies, are cells trafficking there? Do we see any degree of remodeling? Are we fortunate enough to see any engagement of, the innate and adaptive immune system as a function of our intervention? So lots of open research questions that we're beginning to answer translationally that I think help inform our second and third generation products, starting with FT538. So, in terms of an update, 516 is a sort of proof of principle that we can do some of these things. It's not something that we're going to develop beyond phase 1. We're really focused on FT538 and now FT536. And I think similarly, we'll probably try to provide an update around SITC in November again on both programs.
Exciting. So maybe if you could just sort of walk us through Fate's solid tumor strategy, because it's pretty interesting. Sort of combining these standard of care monoclonals with FT538. Can you just walk us through that?
Yeah, so there's 4 different arms. They operate independently. So we're looking at 4 different combinations. The first one is avelumab, different tumor types. Of the PD-L1 inhibitors, it is billed as the most ADCC competent. So we're looking to try to take advantage of that. We also ran that combination with FT5. So avelumab is one arm. There are 2 very well-known antibodies, one trastuzumab and HER2 positive tumors. Good ADCC competency there, as well as with cetuximab. So we're looking at 2 combinations, monoclonal antibodies there. And then the last one is pembrolizumab. We have a strong interest in non-small cell lung cancer. PD-1 inhibitors are used quite often as Keytruda the most in earlier lines of therapy. So we're interested in exploring those combinations predominantly in non-small cell lung cancer. So those are underway. While the first few patients have experienced in different cohorts, and we'll give an update in November. We are really excited about FT536. It takes that same backbone of FT538, and we've introduced a CAR to these stress proteins called MICA, MICB. Yeah, we had FT500, which is sort of an unengineered iPSC cell, and then FT516, which, I think everyone is aware that solid tumors is a much bigger hurdle, a much higher ask. It's a much more challenging set of tumors. So those are initial foray, right? You're trying to answer the questions: can you administer NK cells safely? In the case of FT516, is there any indication that you can combine with monoclonal antibodies, like avelumab or ADCC competent, and see sort of any clinical benefit? Are they safe? A lot of the take-home lessons around translational insights, just understanding from tumor biopsies, are cells trafficking there? Do we see any degree of remodeling? Are we fortunate enough to see any engagement of, the innate and adaptive immune system as a function of our intervention? So lots of open research questions that we're beginning to answer translationally that I think help inform our second and third generation products, starting with FT538. So, in terms of an update, 516 is a sort of proof of principle that we can do some of these things. It's not something that we're going to develop beyond phase 1. We're really focused on FT538 and now FT536. And I think similarly, we'll probably try to provide an update around SITC in November again on both programs.
Exciting. So maybe if you could just sort of walk us through Fate's solid tumor strategy, because it's pretty interesting, sort of combining these standard of care monoclonals with FT538. Can you just walk us through that?
Yeah, so there's 4 different arms. They operate independently. So we're looking at 4 different combinations. The first one is avelumab, different tumor types. Of the PD-L1 inhibitors, it is billed as the most ADCC competent. So we're looking to try to take advantage of that. We also ran that combination with FT516. So avelumab is one arm. There are 2 very well-known antibodies, one trastuzumab and HER2 positive tumors. Good ADCC competency there, as well as with cetuximab. So we're looking at 2 combinations of monoclonal antibodies there. And then the last one is pembrolizumab. We have a strong interest in non-small cell lung cancer. PD-1 inhibitors are used quite often, as Keytruda the most in earlier lines of therapy. So we're interested in exploring those combinations predominantly in non-small cell lung cancer. So those are underway while the first few patients have experienced in different cohorts, and we'll give an update in November. We are really excited about FT536. It takes that same backbone of FT538, and we've introduced a CAR to the stress protein called MICA, MICB. It's relevant in many different tumor types across solid tumors and liquid tumors. And we're in the process of completing manufacturing now. We'll go through the release, and we hope to be in a position by early midsummer to be dosing the first patients in combination with many different monoclonal antibodies. Maybe unlike FT538, there is a monotherapy arm because we have introduced the CAR, so we'll have to get through that first dose level before ungating the combinations. But again, another engineered 4-point edited cell that gives us multi-antigen targeting capabilities in different tumor types across solid tumors.
So maybe we can squeeze into one more about the CAR-T FT819. You guys recently announced that the first dose cohort cleared without any DLTs. I wanted to ask, with all the other CAR-Ts in development, what lessons have you guys learned as you think about how you structure, dosing and consolidation dosing versus single doses? How are you guys thinking about that moving forward?
Yeah, we pay attention quite a bit. I mean, look, this is, well, being hyperbolic, the world's first iPSC-derived T cell. So, many of the same basic questions, is it safe? Is it going to elicit a response? NK cells, we haven't talked much about this. NK cells have been exquisitely safe across many different, therapeutic interventions in different disease areas. T cells, look, we may end up being more similar than different. I will say, though, that we have a lot of novel components in our T-cell therapy, sort of a special CAR, TRK1XX insertion for our CD19. The platform that we have, though, does provide for a much more homogeneous final product, and our ability to do this at scale, manufacture thousands of doses, allow us to play with dose and schedule maybe a little bit differently. So as you look at FT819, there are 3 different disease areas and 3 different dosing schedules, including one that is a fractionated dose. So instead of giving one big bolus, we are now looking at doses on days 1, 3, and 5. That paradigm is not a cell therapy paradigm as much as it is as a T-cell engager paradigm. And the thought there is that you could give smaller doses up front and sort of temper some of the T-cell related phenomenon around CRS and those toxic effects that you typically see. So I can't say that an iPSC T-cell therapy will be like an NK cell therapy and have a great, clean, pristine safety profile. In fact, in many ways, you sort of hope it's sort of toxic at some level because there's activity there, right? So we're running those experiments. We're starting at 90 million cells. I don't think we're going to get anywhere near the 1 billion or 1.5 billion cells that we do with NK cells. So I think we'll learn quite a bit, even in the early and medium dose cohorts. So we're looking at 90 million going to 180 million cells and eventually going to 360 million cells across ALL, CLL, and B-cell lymphoma. And, we'll give an update by the end of the year, probably around ASH for that program as well.
Super exciting. Well, I think we're actually out of time. Ed, thanks so much for joining us. This has been great. I think we're all really looking forward to Fate's, coming milestones this year. So yeah, thank you so much. Thanks to everyone for joining us. Have a good day.